chapter 16. We are on, let's see, page 546. We're going to talk about galactose. Galactose is found in the biological system as a component of the disaccharide lactose, or also known as milk sugar. Now, this is the principal sugar found in milk of most mammals. As you can see here, this is the chain form of glucose, and more specifically D-glucose. And we know that because of the hydroxyl group on the right-hand side. Again, if it was on the left-hand side, it would be L, but it's on the right, so it's D. Most carbohydrates will be in the D form. Now, if we wanted to look at the Howard projections of these, which just means we're determining the alpha and the beta, which indicates of where our hydroxyl and our hydrogen group are going to be on that last carbon. So, on that first carbon. So if it's alpha, the hydrogen's on top and the hydroxyl group's on the bottom. An easy way that I use to remember that is that H comes before O in the alphabet, A comes before B, alpha comes before beta. So if H comes before O, it's going to be alpha because A comes first in the alphabet. If O comes before H, O comes after H in the alphabet, beta comes after alpha, so it's going to be B. It's going to be beta. So if the hydroxyl is on the top, it's beta D, specifically because it was on the right-hand side, beta D galactose. So there is one more sugar in there, and I forgot to write it on the board, but that's ribose. Ribose is a component of many biological important molecules, and more specifically RNA in the body. And on page 547, that shows you the molecule of D-ribose. The hydroxyl group is on the right-hand side again, so it's D. And it also shows you the breakdown if you were going to do the Howard projection. It shows the alpha D-ribose and the beta D-ribose. Now, it also goes into DNA, the molecule that carries the genetic information of cells, contains two deoxide ribose. In this molecule, the OH group on the second carbon, that C2 means it's on the second carbon, has been replaced by hydrogen, hence the design 2-deoxide. And this indicates the absence of oxygen. So if you look on page 547, the bottom diagram, it shows, you see the difference in that, how, well, first off, it's a beta because the OH is on the top on our first carbon. And you see over here that it's the 2 beta D2 deoxyribose. And that, again, is what is found in the DNA. So, on to the reducing sugars. Reducing sugars, the book says that the aldehyde group of aldoses, remember the aldose is the carbohydrates that contain the aldehyde. It's readily oxidized by the Benedict's reagent. And remember that agent is just a basic buffering solution that contains Cu2 ions, which is copper. So that's all that reaction means, is that it's taking the readily oxidized, and remember in aldehyde, the most oxidized carbon on a chain is going to be that top carbon, which is your aldehyde portion, or either your ketone portion. So the copper 2 ion is reduced to just a Cu2, to a Cu positive, which is a basic solution because it's being reduced. Now, this produces a brick red precipitant of copper oxide, Cu2O. And remember, I think in Chem 1000 we did the different, maybe it was Bio 1300, but I think it was Chem, we did the different, the Lepnis paper and the different reactions where we, biology, where we add different elements to molecules. If it becomes that brick red, you know that it's got to be basic and there's got to be copper in there somewhere. So the red indicates powder. So the aldehyde group of the aldose is oxidized to a carboxylic acid to produce carboxylic ion. So this is a breakdown of this, and that's found on page page 548 at the top in chapter 16. So this just shows our aldehyde, and we know this is an aldose, and we know it's an aldose because there's an aldehyde here on the top, right? That's our most oxidized carbon that contains aldehyde, so this is an aldose. If you want to be more specific, there's 3 carbons there, right? So this would be an aldotriose, okay? Because 3 carbons, tri, ose on the end, so aldo for the aldehyde. Now we're adding the Cu2 positive, and we're also going to add hydroxyl ions. And the Cu is the buffer, remember? That's just going to yield our Cu2O, which shows that it's the copper oxide, and it's going to release water when they're combined together.
So you said that's Cu2, is that
Cu2O. And this F is indicating that that's a solid.
Okay.
The precipitant.
I see, so you have it in parentheses. I couldn't make it out.
Yeah, when you have a precipitant, it means that there's something, when you combine 2 things, there's something in the bottom of that test tube.
Okay.
There's some type of precipitant in the bottom.
Okay.
It's not just liquid altogether. Something forms at the bottom, and that's that brick red that you see at the bottom. So, and if somebody wants to stop that, then we'll move to the next thing. Fun, fun, fun.